Hi, my name is Ryan Doss. I'm a second year master's student from Dr. Brian Hedlund's lab at the University of Nevada, Las Vegas. And I'm excited to be here to talk about our research on how we believe these fascinating uracil DNA glycosylase DNA polymerase enzymes may have been a precursor to the extant DNA polymerase A enzymes. And so before I start, I do want to acknowledge all the people that's made this project possible. So first and foremost, Dr. Marika Palmer, who was the postdoc in our lab and is on her way to greener pastures as a associate professor at the University of Manitoba. Um, my PI, of course, Dr. Brian Hedlund, uh, Simon Rue, and the JGI DNA Synthesis Science Program for helping us identify these sequences and helping our collaborators start to express some of them, and our collaborators at Verizymes, led by Dr. David Mead. So I'm going to assume most people here haven't, in their free time, stared at the phylogeny of DNA polymerase, but essentially it's a very complicated uh, phylogeny when you compare it to the tree of life. So every organism needs to replicate its genome at some point. And so early life is believed to be RNA-based life, so it makes sense for those to be RNA polymerases. But then how does it look when we have DNA polymerases? And the answer is very confusing. So you have this is a homologous structure alignment tree. And what you can see here is that viral sequences pervade across almost all families of polymerases. And then across all families of DNA polymerases, we do see eukaryotes in yellow, um, we see archaea in orange, and we see bacterial sequences in pink. So essentially, uh, excuse me, replication occurred pre-Luca, before the differentiation between the eukaryotes, archaea, and bacteria. And because viruses are pervaded across the entire tree, it is believed that gene exchange between cells and viruses during this early node over here help kickstart DNA polymerase synthesis. So I don't have too much time to get into this about replication during the era of the last universal common ancestor. But since a lot of the talks today have been about it, I think if anyone's really interested, this article by Dr. Eugene Kunin in 2020 really drives home what we currently know about replication during that early time period. And so there's very limited homology between the different polymerase domains, or between the polymerase and the three domains of life. And that's believed to be because polymerases are predate even Luca. And so what is led to believe is that RNA-based life has, of course, RNA polymerase, whereas viruses, since they evolve so fast, were and based on the core domains of some of the polymerases being viral-like, it makes sense that at some point they were able to, or that cells were able to recruit viral polymerases and merge them with their own to increase catalytic efficiency. The problem at this point is that these are very rudimentary replication machineries, and they have very low fidelity. So how would cells resolve fidelity issues? And the answer to that is auxiliary domains. So a prime example of an auxiliary domain that most people know of is 3' to 5' exonuclease domains. And so these allow proofreading, essentially, of polymerases to be able to increase their fidelity. Essentially, they make less errors and less mistakes. So an example being Q5, which has a 3' to 5' exonuclease domain and a few other bells and whistles, has over 200 times higher fidelity than Taq polymerase. Much less talked about. In fact, we were only able to find one single paper that shows this, is a uracil DNA glycosylase domain. And so what uracil DNA glycosylase does is it's able to recognize uracils inside genomes and splice it out. And so this is very beneficial in early life when cytosine deamination can cause uracilization inside DNA. And so if early life is is in very extremely hot environments, it makes sense that this could be an earlier adaptation. And so it is found in literature in a bacillus phage called bacillus phage SPO1, and it's shown to have a functional polymerase activity for there. But recently, when we go hunting in our hot springs, we were able to identify it inside a hypothermophile name, or soon to be officially named Furidibacter saccharei. And we were really curious as to how widespread this is. We knew it was found inside that spring, and we knew it would be found in other samples. But where else is it found? And so that search led us here to IMG. So we searched through the IMG-M database at the time, and for proteins that contain a PFAM for both polymerase and uracil DNA glycosylase domains. And what we ended up finding is it's actually really widespread, which really shocked us. We've only seen one paper written about it. We see it in our samples, but we find over 9,000 sequences in IMG's database that shows it. And we find that it's widespread across all different kinds of environments. So we find it inside aquatic environments mostly, but we find it inside terrestrial environments, non-thermophilic environments, which really surprised us. We expected to see it mostly in thermophilic environments due to cytosine deamination. And when we look at a phylogeny of it, and I know it's a big tree, so I'll just explain the important parts of it. The outer rings here represent the environments. So blue stands for the aquatic ones. Orange will stand for the engineered sample, the engineered metagenomes. Pink will stand for the host-associated, and green for the terrestrial ones. And so what we see here is that there's no real pattern of environments that is very clear across this tree. It's all over the place. So it's not just that the the current prevailing hypothesis is that multiple invasions occurred into different environments. And so where are these enzymes found? If we can't make sense of it phylogenetically like that, what can we make sense about it? And so when we look at the scaffolds these genes are found in, they're found mostly on mobile genetic elements. So extrachromosomal linear DNA or circularized DNA found inside metagenomes. So one such example is Furidibacter saccharide here. We do see that there's the uracil DNA, UDG DNA polymerase here. And most of the other things on it is either hypothetical proteins where we don't know what the function is, unfortunately, or DNA interacting proteins. And the idea we had is that perhaps some of these are from viral-like elements. So knowing about SPO1, we used VersortOr2, which is a program hosted here by IMG that allows, that will try to predict whether or not scaffolds are likely to be viral-like. And we found that over 2,000 of about 5,000 sequences were high confidence to be viral-like, which isn't the entire thing, but it's a huge amount. And we do plan on searching in GE-Nomad too for whether or not these might be mobile genetic element-like as well. So when we take it back and we try to relate it back to what we saw with the Montanen figure here, there are two key things that we found that lead us to believe that these are actually from a more basal node than the current extant DNA polymerase A's. So one such example is if you see here, these are where when we map just the palm domain of the polymerases to each other, the DNA polymerase A's are at a much more shallower node than the UDG pol A lineages. They're at a much more basal node. Whereas when we look at the DNA pol B's, we see that they've also belong in the closer towards the root, which leads us to believe that the node that the UDG pol A's come from is somewhere around here. And what's also just really fascinating is that the UDG pol A's almost across the board are at a much more basal node than the canonical pol A sequences. So when we look at what we did here is worried about long branch attractions and other tree reconstruction artifacts. We removed certain groups, certain outgroups from the tree just to make sure that the patterns remain consistent. And for the most part, they seem to remain consistent. We see that the pol A's still cluster at a much shallower node than the UDG pols. We see that the root still tends to group together. There are some problems with reverse transcriptase in general, but we've had that problem consistently where it shows up a bit further down in the tree. And so this leads us to believe that the only conclusion we can really draw from this is that UDG pol A enzymes are derived from a much more basal node than the canonical pol A's, which is really weird for us to think about, but it would make sense for there to be essentially a loss of this domain. Based on this tree, it makes sense that there would be a loss of this domain in the canonical pol A sequences more than this many fusions events that gained the domain. And so what we plan on doing from here is we plan on running GE-Nomad 2 to search for other mobile genetic elements and to also verify that the VersortOr2 results are accurate. We plan on further investigating. So what I didn't have time to bring up today is that there are also UDG pol B fusion enzymes in the tree as well that we're looking at, which can also help us identify where about this basal node is. And then we also are hoping to identify the mechanism that this works. So our collaborators at Verizymes have actually been able to fully express these and show that they have UDG activity and they have polymerase activity. And so what we want to know is why exactly does this work? So UDG is step 1 in the basic excision repair. Polymerases are step 3. So how does that work for repair? Is it potentially viral host warfare since it's found commonly in viruses, or is it a different mechanism entirely? And that's the future direction of our research work. I want to thank you for your time. If there's any questions, I'll feel free to answer it here or at the lunch.
Okay, do we have any questions from the audience?
Hi, Ryan. Really cool talk. I was wondering the alignment that you made for the UDG fusion for pol A, is it truncated or is the fusion outside of the regular pol A domain, and is it like N-terminal, T-terminal? Is it in the middle? And did you have a tree that's inferred with that truncation and the full length, or yeah?
So the question is where exactly is the fusion occurring at, and do we have a tree with and without it? So this tree is just with the palm domains of the polymerase. Because of course, if we were trying to do the tree of the whole gene, of course the UDGs would polymerases would group together. So we cut the UDG part out, so that's at the beginning of the gene. And then what we end up seeing, and then just working with the palm domain. If we look back at this tree is an alignment of all the UDG polymerases to each other. Just so the full gene, not truncated at all. And there is some patterns within them, but nothing in terms of environment or in terms of host or in terms of any sort of evolution structure of life.
Cool. Thank you.
Thank you.
Any other questions? Okay, let's go ahead and thank Ryan one more time.